Vanakkam. Welcome to this video on classics in neuroscience. We have been looking at the DNA, how the structure of DNA discovery led to the explosion of work in this field. In this video, we will be looking at how the DNA works and how RNA was discovered, how RNA is crucial in protein synthesis, and how the code of this replication happens. How does the DNA work? Well, although the structure of the DNA itself was discovered in the 1950s, and although a mechanism for possible replication was presented in the original paper, it was not until much later or until the 1980s this model was clearly confirmed. Also, immediately after this discovery, there was no practical use of this. Immediately after this, there was no practical use or practical effect on the work being done, because this is a groundbreaking work and many more years of hard work was required to make some practical implications from this discovery. In other words, this work of the structure of DNA, or the discovery of the structure of the DNA, was just the beginning, not the end of this line of work. Also, the events of the 1940s and 1950s blurred some boundary lines between traditional disciplines. For example, study of enzymology and nucleic acids in biochemistry. This led to the structure of insulin being explained using a sequence of amino acids. This idea that proteins consist of sequences of amino acids started at around this time. These studies suggested that there is some correlation between the base pairs in the DNA and the sequence of the amino acids in the proteins. This did not answer the things, but rather added more questions to the discussion. This correlation between the structure of the DNA and structure of proteins did not answer the questions that existed, but rather started more questions in this field. Crucially, the following were the questions, because DNA was known to be present in the cell nucleus, whereas protein synthesis itself happens in the cytoplasm, not in the nucleus. So, if DNA is going to dictate how proteins are going to be formed, how is the communication happening from within the nucleus to the rest of the cytoplasm? How is that happening? And there are many such mysteries, right? So, how does DNA undergo replication within the nucleus? So, one is replication of the DNA within the nucleus itself. What is the form of information within the DNA sequence? How is this information reaching the cytoplasm, and how this information is decoded or read out to produce the or to synthesize the proteins? How is this happening? So, within the nucleus you have the DNA, and in the rest of the cytoplasm you have the protein producing structures. How is this information coded, then transmitted, then decoded to produce the or synthesize the proteins? This is the question. These are the set of questions before us. Much before the discovery of the structure of the DNA, people have attempted to study the ribonucleic acid or RNA. These are found in the glandular cells. People started studying this as early as 1900s, and RNA was known to be found in the microsome, rather in the microsome fraction. In the 1950s, electron microscopy was used for these kind of studies. So, by that time people had discovered that there is this organelle within the cell called as endoplasmic reticulum, and there are two kinds of these, smooth endoplasmic reticulum and the rough endoplasmic reticulum, and using radioactively labeled RNA they found some information that could give clues to the discovery of how RNA worked or how it involved in protein synthesis. Crucially, this line of work dramatized or made it dramatic the impact of electron microscopy for this line of work, right. But the question still remained, how does the endoplasmic reticulum, rather the rough endoplasmic reticulum, synthesize proteins? How did it do it? That question was not answered yet. A crucial question is how RNAs help with the synthesis of proteins, as we discussed. In the 1950s, the late 1950s, 1957 or so, Francis Crick gave a talk on the title "On Protein Synthesis" was the title of the talk. So, in that talk he discussed how protein synthesis might happen within the cell, and he discussed some evidence suggesting unidirectional flow of information from the DNA to the formation of protein. So, he suggested two hypotheses. The sequence hypothesis, in which the DNA specifies the sequence of amino acids in the protein. So, the sequence of base pairs in the DNA define the sequence of amino acids in the protein. And also, once information goes from the DNA to the protein, it cannot get out again. This came to be called as the central dogma hypothesis. So, just after the double helical structure of DNA was discovered, it was possible to divide the protein synthesizing mechanism into 4 components. One is an ATP donating component, a soluble enzyme function, a microsomal function, and an amino acid. It seemed like a separate enzyme in the soluble fraction was needed for activating each amino acid, and this led to the identification of the so called transfer RNA by several groups. tRNA. So, the tRNAs contain the triplet code for assembling the amino acids, a triplet code for each amino acid as suggested by Crick. Right. Through ultracentrifugation, ribosomes are the particles that have both RNA and protein identical to those particles that are seen in an electron microscope that somehow appeared to be involved in synthesis of proteins were identified. This of course were named ribosomes, suggesting their content of RNA, and this RNA within the ribosome came to be called as rRNA, or the ribosomal RNA. The ribosomal RNA itself was quite stable chemically. This was not sufficient to explain the whole mechanism, because how information from DNA is reaching the RNA, or how information is getting converted from within the nucleus into the cytoplasm is still not covered. That is when there was this discovery of a less stable form of RNA called as mRNA, referring to messenger RNA. This was discovered, and the role of messenger RNA was to convert the information in the DNA to mRNA, and this came to be called as transcription. And converting the information in the messenger RNA into protein through interactions of the transfer RNA and the messenger RNA came to be called as translation. Okay. So, Crick's predictions in his talk on protein synthesis served as important guideposts in guiding research in this field of protein synthesis for a long time after the talk. Crick suggested in his talk on protein synthesis that information from 4 bases, adenine, cytosine, guanine and thymine, essentially formed the basis for sequencing of amino acids. But how did this happen? That was a bold claim because there was absolutely no data to suggest this. This is where sometimes it is useful to have insights about things that do not exist. So, such was Crick's genius, one would say, to look at the possibility of this becoming true. So, it was a bold claim with absolutely no data. Later, cosmologist George Gamow came up with this idea that the DNAs were indeed a direct template for proteins, and all proteins were made of just 20 amino acids. This line of work also resulted in this explosion of attempts to synthesize RNA or generate a synthetic RNA, more precisely synthetic polyribonucleotides, right. An enzyme that was capable of doing this was identified by Okawa and colleagues, that this enzyme was polynucleotide phosphorylase, and using similar methods, using these kind of methods, people synthesized several proteins, and which was most likely to be made of a triplet of codons, or a triplet of nucleotides that came later to be called as a codon. With this, we come to the end of this video. In this video, we saw how the DNA works and how RNA was discovered, and the crucial role of RNA in protein synthesis, and how the code or the information itself goes from DNA into synthesis of proteins.